spot where it's a nuclear test, and you were green flag. And the way they would check the guys
Okay, folks, if you take your seats, we will get started. We're a few minutes after 10:30. Want to make sure everybody was in the building, in the room. It looks like they are. Now we just need to find our seats. But I'm going to get started with the introduction. By the time that Dr. Tanner steps up, I think we'll be all settled in. So good morning, everyone. Thank you very much for joining us here this cold Berkeley morning. Good for you for making the trek for another in-person event with Pediactive. My name is Todd Morris. I'm the president of the board for Pediactive. Together with the rest of the events committee, Monique Buffler and Carlie Tanner, will be hosting and facilitating for you this morning. Also wanted to reach out and thank our volunteers this morning. You guys can raise your hands. They're helping us check people in. Got a good staff up there. In fact, Dr. Tanner has brought several of her own ringers here, so you'll meet some grad students in the back that she will introduce in her presentation. And then our staff. Can our staff raise their hands, please? Jen? Evangeline couldn't make it today, so Jen's shouldering that burden, but thanks, Jen. So before we introduce the topic, which is titled PPMI Update, Economic Burden and Prevention of Parkinson's Disease. Before we do that, let me give you just a little bit of housekeeping. Sorry, Dr. Tanner, be patient with us. So Pediactive offers all events and movement classes and support groups free of charge in order to get more quality Parkinson-specific programming to more people touched by PD. That includes all 11 support groups, all 12 movement classes, and all roundtables and speaker events like this one. To be able to do this, if you value these sessions, please visit our website and consider becoming a monthly recurring donor. That would be great for us. You can find the big green donate button right on the homepage of pediactive.org. There's also a tip jar in the back, the old school way. For a glance at all of these programs that we're talking about here today, you can check out the calendar, also found on the homepage of pediactive.org. So keep your eyes on that. And a quick word about upcoming events. Do keep your eyes also on your inbox for Pediactive newsletters announcing educational events on topics like a recap of next month's World Parkinson's Congress. Anybody know what World Parkinson's Congress is? End of May, it's going to be in Phoenix, Arizona. Who's going to be there?
Leah.
Ann, Carlie, Dr. Tanner behind me. Pretty good. I will not be there because I have DBS in May.
Oh, yeah.
There's a little brag. I'm so excited. Other topics are ADBS, or adjustable DBS, which is the full circle self-learning model. ADBS and non-motor symptoms, and then pain and PD. Those are some of the topics we're pursuing for some of our next month's events. One important note, this webinar will be recorded, and we will post that recording to our Pediactive YouTube channel in the next few days. As always, the content on our YouTube channel is provided as educational guidance only. Any medical advice on your specific symptoms should be sought from your doctor and who knows your condition best. And we'll send out the link to that YouTube channel when it's put up. Following Dr. Tanner's presentation, we will have ample time for questions and answers. So we'll probably take a very brief little respite between the presentation and the Q&A. For those that need to escape, please do so lickety-splickety, and we'll get back to the Q&A. Good? All right, what else here? All right, let's move on to the real show. This may be one of the easiest and funnest introductions we've done here for the events committee. Some of you may know her as UCSF's co-principal investigator in the Parkinson's Progression Marker Initiative, or PPMI. Anybody here participating in PPMI? That's a good show. Awesome. Or as renowned neurologist and professor of neurology at the University of California, San Francisco, UCSF, Wild Institute for Neurosciences. But sparing you her plethora of accolades and all those letters after every brilliant person's name, I believe this is Dr. Tanner's third time speaking to our group. That's a record. But so there are others with two, though, so we need her to keep coming back to keep her lead. All right, we bring her back for us. So we at Pediactive know her best as Carlie. So without any further ado, here's Carlie Tanner, everyone.
working? Can you actually hear me? Great. Okay. So it's so nice to be here and see old friends and meet new friends, and really lovely to see people who are part of the team, because all that we do in research is teamwork, and you're the most important part of that team because we couldn't do it without you. So thank you so much for everything that you do, and if you're not actually involved, for your support of other people who are. It's all really, really important. It takes a village. So I was asked to talk about PPMI when people asked me to come and speak with you, and I took the opportunity to add in a few other little things, but mostly I'm going to talk about PPMI and give you an update on that. And I know we've already kind of called out our two UCSF team member volunteers who are also researchers, Lynn and Lizzie in the back. And they have been really integral to some of the work that I'm presenting here as well. They're involved in PPMI, they're involved in some of the other work. So again, it's a huge team, and I'm just talking about it on behalf of many, many, many people. So these are disclosures. None of them have anything to do with what I'm saying in terms of conflict. And then, so today, as I said, I'll talk about PPMI. Then I'm going to talk a little bit about a new report you might have heard about that we've been working on for a couple of years, looking at the costs of Parkinson's disease in the US. This is an update. And then I'll touch lightly on prevention because I'm a public health professional, and that is like dearest to my heart is prevention. So what is PPMI? PPMI is an observational, meaning just looking at people, not giving them a treatment. Longitudinal, meaning looking over time. International, multiple centers study. And our aim is to identify clinical, digital, imaging, biologic, and genetic markers of Parkinson's progression to be able to do a better job of clinical trials, especially focusing on slowing or preventing Parkinson's disability. So that remains the holy grail for us. I've been doing this now for many decades. And when I first came in as a fellow, as a young neurologist, we were doing our very first trial to try to slow progression in Parkinson's. We're still doing trials trying to slow progression in Parkinson's. We think we're closer, but we haven't actually gotten there yet. So very, very important. And that's actually what prompted us back before 2010 to set up PPMI. We have little sort of think tanks. What can we do? What is missing? Why aren't we succeeding in this? And came up with this idea of PPMI. So some really unique things about PPMI, and especially in 2010 from a scientific perspective, what's unique about them. And I'll just mention, I'm going to share these slides so everybody can get them, so you don't have to, you can, but you don't have to take pictures. So one of the unique things was setting up standardized ways of doing things that could be shared, right? So lots of studies do standardized methods, but sharing methods so that anybody can then say, oh, I want to do it exactly that way. So I'm going to look at the method they use in PPMI, and I'm going to do that too. This was something new. And the advantage of it is that, let's say, some people over here are doing research, some people over here are doing research, both have sort of small groups. If we combine them, then we're going to be better able to understand what we're trying to test, what we're trying to understand. But combining things sometimes is really challenging because I called men, and you called men, males, and so before I can put those two data points together, I got to recode one or the other, right? So we decided to do standardized methods, make them available to everybody for all these different aspects of what we're doing in PPMI. Then we decided also to make de-identified data open to any scientist to be able to use. So people from all around the world, whether they're involved in our study or not involved in our study, can be credentialed, go to the PPMI website, download the data, and do research evaluations on that information. So this, at the time, was really revolutionary because people who are doing research studies have to protect their jobs, they have to protect their next grant application, and keeping track of your own data is one of the ways that traditionally people would do that. So saying we're making open source data, we're sharing our data, we're sharing our samples to anybody who has a good idea once they follow certain credentials and we know they're real, was really, really revolutionary, and it has made it possible, I think, for lots of understandings to happen that might not have if we just kept it to ourselves. Even if we're smart, nobody has that much time, right? So this is PPMI, started in 2010. We set out really in a clinic. We set out to study mostly people with newly diagnosed Parkinson's. We added in some people who were similar but didn't have Parkinson's or other diseases that we called healthy controls. I'm sorry, that's so tiny. We had 64 people who, when we did dopamine transporter imaging or DAT scans, looked like Parkinson's, didn't have dopamine transporter imaging deficits, so they were called subjects without evidence of dopamine deficiency or SWEDs. We kept following them anyway to see what happened to them. And then we started enrolling people who we thought might be on the way to Parkinson's but not have it yet. So people we called prodromal, before the time of diagnosis of Parkinson's. And so that's the primary group, and then we were focusing also on some of the gene mutations that were becoming identified as associated. So the LRRK2 gene, the GBA gene, and the alpha-synuclein gene around the world, pulling in people with these relatively uncommon, but possibly really informative genes associated with Parkinson's. And so we started in 2010. By 2019, we'd followed people for 15 or more years. Everybody came in regularly. Some of you might have been part of that group. You came in, you got tested, you got blood tests, you got lumbar punctures, you got DAT scans, you got MRI scans, you got all kinds of other stuff. And everybody did it very consistently, so that we had, a huge data set that helped us to start to understand more things. Samples were banked and able to be studied. So by 2019, we had 5.5 million data downloads from people all around the world looking for scientific studies, more than 200 requests to study samples from the biorepository, and then lots of other smaller studies that were happening. Okay. We said, this is interesting, but from what we're learning, we need to go further. So in 2020, what we did is we said, let's do a whole next round of PPMI. In the next round of PPMI, we're going to try to focus much more on that very early stage prior to the time of diagnosis to see if we can identify people who may be at risk, but before they actually get a diagnosis of Parkinson's. And so we took advantage of a lot of new technology that had developed over that period of time and started with online and remote ways of looking at populations to try to hone in on people who might be people at risk for developing Parkinson's. So we started with the idea of PPMI Online, which our team here runs, and then PPMI Remote, which I'll talk about in a minute, but then people got remote smell tests or remote blood tests for specific genes, and then narrowed down the people who were invited into clinic to be people who were most eligible for those, based on those things. So PPMI Online I'll talk about just a little bit because it's one of the things we do here at UCSF. And our objective here was to use online resources to sign people up and collect information every 3 months. So I hope everybody's in PPMI Online because everybody who's 18 or older can be in PPMI Online. And every 3 months we ask you to tell us stuff. And we can put that all together and understand more about the trajectory if you're somebody with Parkinson's, if you're somebody at possible risk for Parkinson's, or if you're neither of those things, we have a strong comparison population of people who are the same age, the same sex, but don't have anything, right? And that's really important for us to know what really matters in identifying people who may be at risk for Parkinson's. And so that's what PPMI Online does. We launched it in about 2020. And just to emphasize, we've done some iterations of it, which I'll tell you about in a minute in terms of how we do it. But basically, being in PPMI now is not being in clinic. It's anybody can be in PPMI, and everybody is important to contribute to PPMI. We now have a new, what we call a portal, so a new sort of one-stop shop for how people can get into PPMI. So you go to what's called My PPMI now, you set up your personal profile, and then you have opportunities available to you. And one of those opportunities is PPMI Online. So you don't have to do that, but we would like you to do that. You go there through the My PPMI portal, which means if you do PPMI Online and then you do something else, you don't have to keep putting in all the data, like who are you and how old are you and, you know, what's your sex and all that stuff, because it's all in that personal profile that you've done. If you change something in one of the related studies, it back changes the personal profile. So it minimizes the burden for people who are getting questions from the clinic and questions from PPMI Online and questions from something else, right? So everything is now linked and together. So that's something we've just started in the last year or so. The other advantage of it is for people who are in clinic, for example, who are interested in having return of their research results. This is the way you get it. So you sign up in My PPMI, you're identified in your personal profile, and then you have the opportunity to opt in and say, I would like to know about my personal research results, and you can understand them there. This is another innovation we've started in the last several years within PPMI, recognizing that this is a partnership and that the old-fashioned way of only the researchers know anything about the people in the research study is really backward and not the way to do this. So now we have a way of letting you know if you want to know it. For some of the things like the genetics, there are counseling available if you want that before you even decide you want to know it or not. But basically this is available for everybody who's in clinical PPMI. Long term, we hope to have it available for everybody, but it's sort of having the capacity and the time to make it available for people who are just in online, for example. So that's examples of some of the other opportunities that you can get through My PPMI in addition to PPMI Online. There are other opportunities we plan for down the road that might be of interest as well. Right now, for people who are in clinic, we're doing cognitive testing using a computerized battery that's been set up from folks in Cambridge. We may be extending that out to other people as well, and that could be available through PPMI Online. So lots of things to come, but this is the place to go for that. So just as a timeline for PPMI Online itself, so we started in 2021, just after we launched our whole big new initiative, and started on a slightly different platform and then migrated over to the one I've just described to you, starting at the beginning of last year. It started out being only in the US, and now we're expanding internationally as well. One of the limits of our last platform is they weren't able to be international. So now we're international. We can comply with the rules that, for example, the European Union has different rules for online data than the US does or Canada does. So now we have all the PPMI sites coming in and being able to participate in PPMI Online. So here's where we are in 2026. We have 7,755 people who have ever done anything in clinic. It's probably hard to see this, but the orange bars, these are just showing visits. So this first visit is baseline, and this is looking at the visits that happen every 6 to 12 months over time. Orange is people with Parkinson's. This is people who are possibly prodromal, so they have certain risk factors. And then this dark color is the healthy controls, right? Oops, there we go. And then this is just looking at the age distribution with those same colors representing the group. So most people are in this 60 to 69 range, 50 to 59, 70 to 79, but some people are more than 90 years old, and some people, these would be people with some of the Parkinson's associated gene mutations, are 30 to 39. We're doing really well in terms of representation of women and men, about 50-50. We're not doing as well as we would like to in terms of racial distribution. It really doesn't reflect the whole population, and we have many more white people than Asian or Black or Pacific Islander, American Indian, or multi-race people. So we still have work to do in that regard to be more representative. This is a big schematic, and trust me, I'm not going to talk you through it, but I just wanted you to get the picture of what PPMI is, because each one of these, each one of these little boxes represents a team of researchers that's probably anywhere between 4 to 25 or 30, working on a particular aspect of a project of PPMI. All of the remote opportunities feed through the My PPMI portal, and people in PPMI sites also connect to the My PPMI portal. But then we have all these other aspects for managing the sites, managing the data, collecting the imaging, taking care of the biologic samples, screening and doing and reporting the genetic testing. People in PPMI are kind enough to actually donate their brains when they pass away. So we've also been able to learn a lot from being able to do standardized research evaluations of people's brains. If they were in PPMI, it's almost unprecedented to know that much about someone before they die and then be able to understand more when we look at their brain pathology. And then all the people who do the analyses, who take care of the statistics and the bioinformatics, the analyses of the different imaging samples and blood samples. And who takes care of this overall? We have a steering committee, an executive steering committee, the Fox Foundation, who are our partners in this all the way through and are not just helpful in terms of the scientists who are in the foundation being involved, but helpful in terms of making this a public-private partnership so that the support for PPMI primarily comes from private fund resources. It's not government funded. That's why we could do it, frankly, for this many years, because NIH grants us 4 years or 5 years, and then you have to try to get refunded, and in the process, you usually lose a lot of samples. So the Fox Foundation gave us that support for funding and continuity, and they did it by partnering with other foundations and other private donors, but also with industry. But their caveat to industry was that this is pretty competitive, right? So no company could say, I'm giving you $1 million and I'm going to own part of PPMI. The company could say, I'm giving you $1 million because PPMI is important, and my scientists will have the same data access as somebody around the world who doesn't have any relationship to PPMI at all and wants to pull down the data and do an analysis. People in industry are our advisors. They recognize the importance of this pre-competitive space to collect information that ultimately helps them do a better job of designing their clinical trials and designing their research. But nobody in any company is going to own PPMI. So that too was revolutionary, and thanks to the visions of the Fox Foundation for making that happen. That's why we're still here today, all being part of PPMI. And then there is a scientific advisory board that is the community advisory board, which includes people from various industry companies, private donors, people like that. And then we have a very important patient committee who also are advisors to us, who keep us honest and make sure we're in the right direction and give really valuable input at every point to the work that we're doing. So that's the structure of PPMI. It's been really helpful. So that's swell, right? But have you learned anything? So one thing that I'm going to talk about is one of the earliest things we wanted to do was identify people at risk or in the earliest stage of disease, because we think this could be the best time to intervene to slow or stop progression. So this little diagram shows all those little boxes are symptoms that people with Parkinson's experience. This is the time of diagnosis. This is the time of the onset of motor features. But before motor features, for many people, there are other things that are changing, including their sleep. One version of this being rapid eye movement sleep behavior disorder, where people act out their dreams. Another being changes in mood, depression, anxiety. Another being changes in what we call the autonomic nervous system function. One of the most common ways that manifests is constipation. And then loss of sense of smell. People who lose their sense of smell more than other people of the same age and sex may be at greater risk of Parkinson's. So we know those things are there, and we use them, especially focusing here on the example of hyposmia, to try to understand people who may be at greater risk of Parkinson's. So this is a study that was published almost 10 years ago now, that looked at people who had loss of sense of smell, sent them in for dopamine transporter imaging. So this is normal dopamine transporter imaging. This is someone who has an abnormal scan on their way to Parkinson's. So this is the important part of the brain that's less active in people who have Parkinson's. And what we found in the study was that people who had reduced sense of smell were much more likely to have dopamine transporter imaging that looked like this and not like that normal pattern that we would expect to see. And that if you looked over time, they were much more likely to develop Parkinson's. So this, each little step here is people developing Parkinson's, and the people who had the dopamine transporter image or the smell deficit were much more likely to have that happen. So that was a clue, but it wasn't very useful really because it still was pretty non-specific. So we were able to take PPMI and try to say, can we be more systematic about how to do this? So this is work my colleague Ethan Brown was one of the lead analysts on this. We took people in PPMI Online and another community initiative called Smell Test Direct. We asked people to do scratch and sniff smell tests. If people had an abnormal smell test, then we sent them in for a DAT scan, dopamine transporter imaging study, and found that people who had abnormal smell tests were pretty likely to have abnormal DAT scans as well. So it looked like this may be a way we can start to look at people in populations with a simple scratch and sniff test and maybe identify people with dopamine transporter imaging studies. But then another kind of revolutionary finding occurred in the context of PPMI, which is people developed the abnormal seeding assay for a protein called alpha-synuclein. So you probably heard about alpha-synuclein because that's the central protein abnormality that's a problem in people with Parkinson's. But what has been the dilemma is that we hadn't been able to recognize that in living people. We could only see it in people's brains after they die. So we could look at somebody's brain and say, oh yes, we see abnormal alpha-synuclein in the part of the brain that's related to sense of smell, and that relates to people who have pathology related to Parkinson's, but we had no way to see that when somebody was living so that we could say this is a person where we want to intervene and maybe try to slow or stop progression. So when this alpha-synuclein seeding aggregation assay was developed, we were able to take advantage of the persistence of everybody who volunteered for PPMI, who gave us spinal fluid samples that we had banked, and go back to somebody who might have been tested in 2010 or 2012 and look at the sample from them and see if there were abnormalities in alpha-synuclein or not. And what we found was that this CSF test for abnormal alpha-synuclein protein was very sensitive, meaning that it was really well able to recognize people who had Parkinson's disease compared to people who had no Parkinson's, and fairly specific, so there weren't a lot of people who didn't have Parkinson's who had abnormalities on this test. So it looked like a pretty good test for distinguishing people with Parkinson's. That was step one. And then looking at other forms of neurodegenerative Parkinsonism, so progressive supranuclear palsy or corticobasal syndrome, which has a different protein abnormality. It's different than Parkinson's. We could distinguish those people as well. So they were popping up as what we would call a false positive. So it looked pretty good in people who already had established disease. And this is what we could see in people with PPMI. This is a complicated picture, but all the positive spots are the blue ones here. This is the synuclein aggregation assay right here. And these are people who did have Parkinson's and they didn't have a genetic form of Parkinson's. And looking at their sense of smell, with the abnormal being right here, and looking at their dopamine transporter imaging study, with abnormal being below this cross line. And you can see they're all clustering together. So in fact, the abnormal alpha-synuclein is what we found in people who also had the other things we know are related to Parkinson's. So reduced DAT scan and impaired smell. But even more interesting was when we looked at the people in PPMI who didn't have Parkinson's disease or Parkinsonism, but they did have a loss of sense of smell, or they did have REM sleep behavior disorder. They too had abnormalities in alpha-synuclein in those same ways. So this is where we said, wow, we may actually be able to use this biomarker test to identify people at risk much more specifically and flag them as people for intervention to slow or stop disease before they get to the point of that Parkinson's diagnosis. So this is like happening now. This is like in the last couple of years. It's thanks to people who have volunteered in PPMI that we're able to be at this point. So we've actually gotten close to what we started out to do in the beginning. The caveats to it are, it's a spinal fluid test, hard to do a population screen that way. We're looking for a blood test. We're looking for, maybe a skin test. There is a skin test. It's not as specific. So more to come. There may be imaging soon that will help us, but this is a big indication that this will happen. And we'll be able to screen people for this protein abnormality and identify people at risk before they manifest disease, which gives us a whole leg up for treatment. And this is applying the same thing. This is that same pyramid I showed you before, my colleague Ethan again. And what we did is we took that same analysis, and now what we added in is those people who had the abnormal DAT scans. We also looked at their synuclein aggregation assay, and what we found is that if you had an abnormal sense of smell, an abnormal DAT scan, you are very likely to have a synuclein aggregation assay of the type that people would see in someone with Parkinson's disease. So again, one more proof based on the data in PPMI that this is the right direction to be going. The other thing that happened at the same time is that people in other research populations were looking at this aggregation assay as well, and people who were studying dementia with Lewy bodies also found those people had abnormal synuclein aggregation assays of the same type that people with Parkinson's had. So these are two disorders. We knew they both had synuclein pathology. Wasn't completely clear what the relationship was between Parkinson's and dementia with Lewy bodies. And this was followed up by looking at people with dementia with Lewy bodies compared to controls, same sensitivity and specificity pretty much as people with Parkinson's, compared to other Parkinsonian disorders that have a different protein, so PSP or corticobasal, similar sensitivity and specificity as we saw in people with Parkinson's, And then looking at people who had REM sleep behavior disorder, so dream enactment, compared to controls. Not quite as good in every population, but again, looking like these people too might have the same abnormality, or at least some of them, as people with Parkinson's or dementia with Lewy bodies. That led us to this idea that this may be the biomarker for a single disorder related to synuclein pathology that has a lot of different ways of manifesting clinically. So one protein abnormality, different people have different syndromes. So for some people, we're calling it neuronal synuclein disease because that's the protein, and the presence of disease-specific alpha-synuclein pathology, dopaminergic neuronal degeneration, but the different syndromes, Parkinson's in some, which is mostly a movement problem, Lewy body dementia in others, which is more of a cognitive syndrome, other people who may have a more neuropsychiatric abnormality, anxiety, depression, people with REM sleep behavior disorders, so dream enactment and sleep problems, or people with other features like prominent dysautonomia, people with prominent loss of sense of smell. All of those people can have the same biology, but these different clinical manifestations. So this is our hypothesis. It's still a research idea. It's not something that happens yet in the clinic, but it gives us a way to think about how to address disease modification or prevention of the full-blown manifestations of Parkinson's from a research perspective. So this diagram shows you a make-up idea of what we think may be the way that things progress in people with neuronal synuclein disease. Okay, so what we're looking at here are, this is what you can see or detect. This is the change, the red line is the change in synuclein, so that happens first, that's causative, or at least a big part of the cause, and the marker we have. After a little while, this is time, you start seeing changes in the dopamine system that could be detected. And then later on, you actually see changes in people's functions and symptoms, right? So there's a progression that goes over time, and we use this idea to come up with an idea of stages. Stage 0 being the few people with a synuclein gene, stages 1A to 2B being people who have some biomarker changes, maybe some mild clinical features, but nothing that's functionally impairing. And then once you get to stage 3 or more, you also have functional impairment from some of the syndromes that people have. And we're using this idea to say, can we target this point before people have functional impairment, when they have a biomarker change, to intervene to slow or stop progression of neuronal synuclein disease. So this is the point we are at this moment. This is our research question. And again, largely coming from the commitment of people in PPMI and your willingness to give us samples and specimens and imaging over all these years. So I put in my title Precision Medicine, and what is precision medicine? So precision medicine means the right person in the right time, the right treatment for the right person at the right time. So finding what your biologic characteristics and clinical features are at this moment and giving you the treatment that will be effective, not just in helping your symptoms, but hopefully stopping your disease from progressing. And we think that we're going to keep building on what we found with alpha-synuclein, adding in other biomarkers, adding in other things we can measure, so that we will know more precisely where somebody might be in that trajectory, and maybe more than one treatment, or maybe one specific treatment for this stage and a different one for that stage. So those are the research questions we're trying to address. And applying precision medicine means we figure out how you got there, we figure out how you are right at this moment in terms of your clinical state and your biomarkers, and then we look at the molecular changes in your body and give you a treatment that reverses that abnormality or stops it from progressing. This happens now a lot in people with different types of cancers, where they can take a piece of the cancer cell and look at the tissue and say, oh, it's this and this and this, we're going to give these 3 drugs, because that's what's, that's what we see in the cancer. We're not going to take people's brains and chop them up and then decide if the right treatment is right. So we have to sort of triangulate a little bit more than a cancer doctor might, but we're headed in the same direction. And this is the way we're getting there, thanks to people in PPMI. So here's what we hope to start this year, and this is called the Path to Prevention Clinical Trials. It's going to be for people who are in PPMI, who have, right now at least, this is our starting point, who have positive alpha-synuclein biomarkers and abnormal DAT scans, who don't have Parkinson's or dementia with Lewy bodies or some other diagnosis. And we're going to do what's called a platform trial, which is an efficient way to do sort of quicker tests of new treatments. Because as you probably know, most clinical trials, you get a big bunch of people who get the active treatment, a similar bunch of people who get what's called a placebo. That's the way you can be sure that the treatment is having an effect or not, but it means hundreds of people, usually over at least a year, often more than a year, and if you get to the end of it and that wasn't quite right, everything's done, you have to start all over again. It's been sort of an analogy to if you wanted to play a football game, for every game, you build a stadium and then tear it down again and start again at the next game. So that's not very efficient. So a platform trial sets up a control group and then has multiple comparison groups who are also enrolled at the same time. So all the different comparison treatment groups can take advantage of that control population. You can have a smaller control population. If you're a participant, you have better odds of getting one of the study research treatments compared to being in a placebo group. And it's much more efficient as a way to kind of quickly say, is this treatment going to work or not? Or if not, let's move on to the next one, right? So that's what we hope to start probably by the end of this year in PPMI. So we'll have time for questions later. If there's a burning question now, I could take it, or else I could just keep telling you about the other couple of things I wanted to mention. Okay, right. So we've just finished this work, and we just presented it actually in Washington. It was released in February, and I thought it was worth mentioning to this group because I think it's also really, really relevant to people with Parkinson's and those in that community, in our community. So what we did was we looked with the help of a group who are sort of experts in doing cost analyses of health utilization data and determined the cost of Parkinson's. But importantly, we didn't just look at direct medical costs, that is, how much does the doctor cost, how much does the drug cost, how much does it cost to be in the hospital. We looked at the out-of-pocket costs that people reported to us. We did a survey in Fox Insight, which is another online forum that our team also manages, and asked people to tell us about their expenses. We looked at non-medical costs, so things like having to change your home so that you could have access to rooms if you had mobility problems, things like that. We looked at the cost of disability benefits, and we looked at the effects for both care partners and people with Parkinson's of losing their contributions to work, losing their contributions as volunteers, losing their other societal input, all of those costs that don't usually get counted, but are really, so critical to our whole community, the loss of contribution from people because of the burden of Parkinson's. And note that the direct medical costs of this green part here, so this is a lot of money, it's $23.8 billion, right? But the indirect costs are 26.2, and I will say that I was one of the advisors on this. They only let me ask about 2 care partners. And I said, it's more than 2 for most people, so we really should be broader, but we just couldn't do it this time, maybe next time. So this is the indirect cost, looking at the person with Parkinson's and 2 care partners, close persons to that person who may have needed to change what they do in some way because of the person having Parkinson's. Disability benefits are 8.3, non-medical costs 18.2, and then all those other things that are not reimbursed by anybody, $5.7 billion in 2024 only in the US. And only for the people we knew had Parkinson's, not taking into account that there are probably a fair number of people who haven't been able to get access to care, who may not be well represented in this sample. So it's likely more than that for many, many reasons. So this is important to know because it is a huge burden. And I think for us, for advocacy, for sharing this information, and for recognizing the impact on the community, this is really important information. And then this is a projection that we made. So this is now, 2025, we projected $84.4 billion. By 2045, $112.4 billion. I will say we did an analysis like this maybe six or 8 years ago, and by the time we started this analysis, we had well exceeded what our projected costs were for 2030 based on that analysis. So again, these are projections, but if things are changing, as you've probably heard, there's at least the idea that perhaps the numbers of people with Parkinson's are increasing. This could go even higher. This is a very conservative projection. So what does this have to do with prevention? I'll just mention a little bit about prevention as a public health person again. We think of primary prevention, which is what most people think of, which is the disease never happens, right? You get rid of what's going to cause it and somehow you block it so nothing happens. Secondary prevention, though, is preventing the onset of symptoms. So people who have that biomarker, they have something going on, can we keep them from getting disease? And then tertiary prevention is, okay, you do have some symptoms. What can we do to slow or stop progression so you stay the way you are now and don't get any worse? So there are 3 phases of it. All of them are prevention. All of them are important, and all are really high priorities for our research. So we'll start with the idea of primary prevention, because that's kind of in the way we would love that to be the case, no more Parkinson's. I'm not going to go into detail about this because you've had a lot of talks on this, I know, but basically genes and environment for almost everybody are contributors to the cause of Parkinson's, and there are many different factors. And this is a schematic. I made it up. It's not exactly based on data. It's just kind of my opinion, but I think it's pretty close to what's probably true. Most people are purple, meaning they have lots of different determinants, 1 or more genes, several different environmental factors. Some make them more risk, some make them lower risk. And then there are a few people who have maybe just a single gene. People with the alpha-synuclein gene abnormality, for example, are pretty likely to get Parkinson's. After that, most people have multiple factors. So that's just a schematic to talk about the different causes. And then this is something based on some work we did maybe 10 years ago, looking at a population of farmers and their wives who were pesticide applicators, and we were able to measure their exposure because they had to keep farm records. And this is called the Agricultural Health Study. We looked for Parkinson's in the Agricultural Health Study, which was originally set up to study cancer risk. Again, back to cancer. And we were able to take advantage of that. And here's what we found. We found, first of all, that a couple of pesticides, one of them being the herbicide paraquat, were associated with an increased risk of Parkinson's before we just looked at everybody. Then we started parsing it out. And one of the questions we asked is, what about diet? We've had this mind that a healthy diet, an antioxidant diet, a diet sort of like the Mediterranean diet, may lower disease risk or may make people less likely to get Parkinson's. And so we looked in this population and said if people followed a healthy diet, but they were exposed to paraquat, what happened to their risk of disease? And maybe there was a tiny increase, but it was what we call not statistically significant, so we couldn't be certain that it was really increased. But if we looked at people with the converse, an unhealthy diet, high in animal fat, high inflammatory potential, and they used paraquat, they have a 4.5 times greater risk of Parkinson's than the rest of the population, suggesting that maybe you could prevent Parkinson's if you were careful about some of your behaviors like diet. Another thing we did, and this is just a sort of a proof of principle almost, is we looked at people who were mixing and applying paraquat, and we looked at those who used healthy protective methods. So they used, they washed off, they used gloves and masks, things like that, compared to people who did not do this. And what we found is that people who did not do this, again, were about 4-fold increased risk of Parkinson's, and people who did use healthy methods had a borderline, not statistically significant increase compared to people who weren't exposed to paraquat. So, again, two kind of proof of principle in very small populations that suggest maybe even in this setting where there's a pretty high risk if you have this exposure, you can prevent Parkinson's, and that has led to some advocacy work you might have heard about, looking to control the use of paraquat in the U.S. that many of the foundations have been involved in recently as well. This is getting back to the idea of other ways of slowing or preventing disease, and I just like to show this slide. It's a little bit old, but there are more things that we could put on it now. But basically, this just shows looking for people who have disease, looking for people who don't yet have disease but may be at risk. There are lots and lots of treatments that are being investigated in different phases. Phase 1 is the earliest, phase 3 is the latest and closest to possibly being approved. And these are, top half is disease modified, meaning things that may slow or stop progression. Bottom half is symptomatic therapies. So a lot going on. Just the visual, I think, is helpful. And then just to end with a few quotes from Michael J. Fox, who is the reason for all of this fundamentally. When Michael got Parkinson's, he used that as an opportunity to set up the Fox Foundation. And the Fox Foundation started out lean and mean, and I was part of the early team, where we would have like $50,000 or $100,000, and what can we spend? And we wanted to de-risk wild and crazy ideas, and that's sort of like where it started. And now it's like this huge thing that supports things like PPMI and all kinds of really important things as well. But still with that same idea of being flexible and agile and not being bound by convention, but always looking critically at what is happening and is this the right thing or should we be changing it? So this is a quote from Michael, which I think is so relevant, that the patients are the experts and you have a responsibility to share your experience, to let us know, to try to keep the researchers on track, if you will, because if we don't have the input from you, if we don't understand what's important to you or what your experience is, we're not going to be able to do our job well. So that's one important quote from Michael. This will also be, like, there's a brochure back there and also be in the slide set. UCSF MOVE trials is the different clinical trials at UCSF, and you can connect into what we're doing. The other code is to sign up for my PPMI. Once you do that, you can be in PPMI online, and we would be most grateful to you if you do that, if you're not already doing it. And then I just want to thank everyone, our volunteers, our research partners, patients, controls, family, friends, and all the funders who are at the bottom. And then I'll be happy to take any questions.